Good evening, everyone. This is another opportunity to meet at eduinteract.ai.org. My name remains Bio-Edutainment Michael, and this evening we are going to talk on microbial physiology and metabolism. So, what are they? In this presentation, we are going to look at the differences between the two, and that one is going to give us a better understanding of exactly what should be expected in this course. Don't forget that this course is for microbiology of 100 level, and one way or the other, there's no way. So at this level, it is expected that everybody should be able, a microbiologist should be able to know at least how to draw a typical um, bacterial cell. So if you are able to know how to draw that typical bacterial cell, you should be able to differentiate between eukaryotic cells and as well as prokaryotic cells. Don't forget that in one of the courses I taught you earlier on, I was able to give you some of these distinctions. But whatever or whichever way we want to let it go, I want you to let this sink into your intellect that at least this time around, you should look for a typical bacterial cell as a cell uh, that is just like a factory, a complete factory, whereby you have raw materials, you have production, you have outputs and input and so on and so forth. So looking at what we have today, is going to be the system biology blueprint. We want to look at what are the cellular processes, the energy pathways, and microbial adaptation. So it's like we want to study the totality of all the activities that are found in the cell, including the way and manner in which a typical cell is able to adapt to its environment. Don't forget that in ecology, microbial ecology and evolution, I actually mentioned that the survival or the survivability of any organism will depend on how they are able to interact with the environment. So that will actually guide us a bit. So looking at this diagram, I want you to pay particular emphasis to the following. I want you to look at this is the nucleus. This is typical nucleus. But don't forget that the question that you should be asking now is that when we are talking of bacterial cell, do we have nucleus in there? So this is a typical, just to illustrate, but don't forget that there are no organelles in typical bacterial cell, no organelles, no localization, no localization of organelles. You cannot see any distinct nucleus. What you can just see is that, and don't forget that the DNA of typical bacterial cell is circular. So, but what I just want you to realize from here is just that this is a typical cell wall. You should know it is very, very important for the survival of any organism, especially bacteria. The cell wall plays pivotal role in it's either making the organism to be able to interact with the environment, because materials go in and come out through the cell wall, and as you can see, ATP is entering, and that's why you have inside this cell you have the glycolytic enzymes, and outside have a kind of ribosome and some other things are coming in. So without wasting our time, let's go to understanding the major concepts of microbial physiology and metabolism. What do we mean? We are talking of microbial physiology, and what are those things that is actually expected if we are talking of microbial physiology as well. Don't forget, this presentation is based on the Edu-Interact AI framework, as is being anchored by Adeotola today. Michael is my name once again. Don't forget. So metabolism, I want you to imagine there are two main parts of this course or of any microorganism. The first part is microbial metabolism. What do we mean when we say microbial metabolism? Number one, we are regarding microbial metabolism as the engine of that cell. No vehicle can move. No vehicle can move without the engine. So if you are able to imagine the metabolism, as we have to be talking about here, as engine, you understand it. So all the cellular activities in the cell is regarded as metabolism. So that's why we said the microbial metabolism is talking about the engine of the cell, and it focuses on biochemical pathways. Which has to do with catabolism and anabolism. Don't forget that anabolism is the breaking down, and catabolism is the making up. All this, I'm very sure you must have been taught in microbial biochemistry. So I want you to go back, revise your knowledge about it, because without microbial biochemistry, you might not actually, because it is at that level you will learn a lot about catabolism and anabolism. You have to talk about building up and breaking down. So having said that, then you will now know that whenever we are talking of microbial metabolism, we are as well regarding as isolated chemical reactions, and how energy are being transferred. So that is that. Then it is also a subcomponent of a broader biological system. So that means without metabolism, without microbial metabolism, the totality of that organism is null and void. That is, it is not going to, without engine, no vehicle can move around without engine. Engine is going to be a kind of propelling force, that will be able to push forward and be able to make sure that your engine, which is the whole of microorganism, is going to move. So what we are trying to say in essence is just that whenever we are talking about biochemical pathways, including the building up and the breaking down, we are one way or the other referring to the metabolism. So you can imagine metabolism to be the totality of all cellular activities or microbial cellular activities, combine them together, whether it is a building or whether it is a breaking it down. So that is it, because without these activities, cellular activities, there won't be any propelling force that will be able to push forward the whole. But don't forget that if any microorganism is to survive in an environment, that means it is going to involve building up of some, or taking some chemical reaction will take place. For example, just like whenever you have antibiotics, if suddenly you now have antibiotic under normal circumstance, antibiotic is going to be too toxic to them and it's going to kill them off. So the only way that organism must survive and will be able to survive is to alter its cellular activity. It's going to change. It's not going to be stationary. It's not going to be static. So that is the reason why. That means for microorganisms to be able to survive, in the presence of antibiotics, they will have to adjust. The engine will be adjusted in order to make them to be able to survive in that environment. So that is that about microbial metabolism. So even if you don't know how to describe metabolism, just describe it as the totality of all cellular activities, including breaking up and building up. That is all chemical reactions that takes place in an organism. That is talking about microbial metabolism. So the next one is microbial physiology. Why do we continue to work? Why will a cell continue to be dynamic? Why will cell not be static? They will not be static in order to perform certain functions. This function can be respiration, it can be reproduction, it can. So what we are saying in essence is this totality of all the activities within that cell will now lead them to where. So if you have engine inside your car, there will be movement, and that means you can move from one place to another. So the same thing, moving from one place to another, that is a function, that is an action. So it is the engine that will determine the action. It is the metabolism that will determine the function. It is the metabolism that will determine the functionality. It is the metabolism that will tell us whether the organism will be able to cope with the environment. So that is why you have to understand. If you do not understand these two distinctions, you might find it so confusing that you might not be able to know the difference between metabolism and physiology. So microbial physiology is talking about the survival strategy. The survival strategy, and one of the survival strategy is their ability to acquire enough energy that is going to help them to be able to grow, to be able to reproduce, to be able to respire, and to be able to function as well. So you can see that the function of any organism will depend on the metabolic activity of that organism. So because that is what is going to help them to whether they are going to survive or not. If no cell divide, if there is no reproduction, that cell will just die off. That means the cell will not be able to cope, the cell will not be able to survive. So alteration of the genetic or alteration of cellular activity in order to suit their present situation will depend solely on the metabolism, so to say. So, and it also includes adaptation to the environment as well as signaling depend on. So under normal circumstance, before the cell will divide, not only divide, before the cell will carry out any cellular activity, something will trigger it. A signal must be processed before it will be carried out. So that is what another thing that is going to dictate. It is microbial physiology, physiological processes that dictate the genetic regulation and the structural integrity. It's going to be the one that will preserve whether to acquire. For example, now take for example the case of Mycobacterium tuberculosis. So once they enter into the lungs, immediately macrophages will be deployed. The first line of defense will be deployed to go and swallow up. They are professional phagocytes. They are very professional. Whatever comes on their way, they just swallow it. So, but one thing is just that immediately the microorganism gets into the macrophages, it's not going to struggle with macrophages that please don't swallow me. No, it's not going to struggle with it. The macrophage will perform its own function, so it's going to swallow. But the next thing you see microorganism, that is the bacteria, doing is to go and acquire. Uh, so I'm trying to talk about the structural integrity. It's going to acquire an additional mycolic acid that's going to be protective, that will prevent the immunological apparatus of the host to be able to harm them. They will not be able to be digested because they have already altered their external host. So what we are saying in essence is microbial metabolism. Yes, the survivability of any organism will depend solely on the metabolic pathway or the metabolic cellular activity. So one thing I want you to take away from here, from this slide, is that number one, how does a microscopic self-contained biochemical factory, how do they survive? How do they adapt? Then how do they thrive in different environments? So whatever we are, what we are trying to say is just that the continuity of any life will depend on their ability to be able to adjust their metabolic processes, as well as to be able to function in whatever environment that they find themselves. So that should be established as well. But don't forget, as a takeaway, that microbial metabolism is going to talk about the engine. Imagine it as an engine, and you should imagine microbial physiology as what? The function. Here, okay, we will see it in the next slide. Okay, this next slide is talking to me about the thermodynamic open system. So some of us might just be wondering, thermodynamics is being, because we are already mentioning factory, already mentioning um, factory, we are mentioning biochemical pathways. So that is to actually tell you us that chemical reactions will take place. And don't forget that one of the law of motion is that a body uh, body will remain in a state of stationary, in the stationary phase, unless it is being acted upon by external force. So what are these external forces? The external forces are the environment, the environmental conditions. That is how well. So what we are saying in essence is just that the survival of any organism will depend on their ability to cope, to acquire the necessary energy from the environment, and the ability to also dissipate some of this energy. So what we are saying in essence is just that no energy should be wasted. The energy can only be converted from one form to another. You can see that matter and energy inputs is coming towards it. And don't forget what we call entropy in chemistry. And entropy is just the internal, it's talking about the internal reaction. So you will see that as soon as this one is just coming in, there is going to be energy coupling. Energy coupling means that there will be adjustments within the system, adjustment, so that this organism is going to be able to cope even in unfavorable condition. So, and if they are able to cope in that unfavorable condition, they will not be so selfish. The next thing is to make sure that they dissipate this energy in form of waste, in form of releasing the ATP, which is the energy currency. It is going to continue. So ATP that is being produced, they are not going to be selfish. If they acquire 30 ATP, they must look for a way of using that ATP, the energy they get, which is the energy currency. They will. So the ATP is just like the cell naira, their own currency. That's why they call it currency. So they will not go and accumulate. So they will not accumulate the currency that is ATP just for accumulation sake. So the next thing is that they will be able to, although they will have to store some, but majority of it will be expended on what, on into the environment in form of energy release. So this side, there are going to be one side that is going to fulfill the energy, thermodynamics law, that microbes must continuously exchange energy with their environment, and energy is not created just the same way that the thermodynamics law, that energy can neither be created nor destroyed, but it can be converted from one form to another. So the same way, so entropy, which is the what is going to happen here, the energy this place is fulfilling, yeah, ability to survive in unfavorable condition. So they will acquire energy, and the next thing you see, they will use that energy to be able to drive their electromotive force. They will be able to drive their cellular activities. They will be able to use it to perform certain functions. They will be able to use it for their structural integrity. And the next one is that all this energy will not be wasted. It will be dissipated into the environment. So that is how you can relate first law of thermodynamics with the open system of a bacteria. Okay, now looking at the cytoplasmic membrane, don't forget that the cytoplasmic membrane, cellular membrane or cell membrane, is made up of lipid bilayer. Lipid bilayer. So you can see this is one layer, which is the upper layer, and this is the, so this upper one is the milieu, the exterior layer, the exterior phase, but this one is the cytoplasmic matrix. So when you look at the number of positive to the number of negative here, electrons are released from here, and the protons are found outside, that is the exterior. So the concentration, the number of these will depend on the direction in which there is going to be movement. If the environment outside the bacterial cell is overly charged, that is positively charged, so in order for that organism to be able to survive, there must be a point of equilibrium. That point of equilibrium is that the number, and that is the state at which all systems must continue to exist. So the number of positive here must be equal. That is the ultimate goal of every organism, to be able to make sure that the number of positively charged outside is equal and opposite to that of the number that will be positively charged at the cytoplasmic matrix. So don't forget our osmosis, diffusion, and the energy. Yes, okay. So just like I said, you will see this is a positive. You can now see that this electron is coming outside. Don't forget, this is this type of protein is called the intrinsic protein. It's called the intrinsic protein because it transfers between the upper layer and the lipid bilayer. You can see it here. Okay, so the core concept is that the structure enables function. I told you earlier on that the structure of anything in life, the structure of anything, be it organism, determines the function it's going to be. So the membrane is an electrochemical reactor that assists the electron transport system. So you can see the way electron is being exchanged. Interior, we have low hydrogen concentration interior, because you can see most of them, all of them, they are negatively charged. So hydrogen will be, more of hydrogen will be driven from outside to the inside. So that's why the stereo very high in hydrogen, very high in hydrogen. So and if the stereo is very high in hydrogen, that means movement of that proton, which is hydrogen, will be more towards the internal, towards the uh, cytoplasmic matrix. So this proton motive force act as stored electrochemical energy, powering flagellar motility, active transport. That was what I wanted to say the other time. Active transport and ATP synthesis. So the sum of the electrical and chemical. So what we are saying in essence is just that the cytoplasmic matrix, you can also imagine it as a battery, a battery, a battery that will have both positive and negative. So and what determines the flow of energy depends on, because every cell will be looking for of how to survive in any environment at all, including the environment that is also very very unfavorable to them, that is so toxic to them. They have to look for a way of how they will be able to cope and live and survive in that. Okay, the other part is just that what do we understand when we talk about the chemiosmotic theory? I've just mentioned something about osmosis the other time. It has to do with the movement of electron wherever. So now imagine it, the electro the molecular turbine is just like a protein which transfers between the between the lipid bilayer, and you can see this one is F1 and this one is F0. F0 because the you can see the movement of hydrogen, just like I said, of hydrogen atom uh, that is positively charged is towards the interior, and it is at that point in time as it is coming in, the positive will be used to liberate uh, hydrogen sorry adenosine triphosphate, that is ATP. Now let us see what happens. Step 1, electron flow. ATC protein transfer electron from NADH. So I want you to so as not to be too confusing for you, I want you to first of all go back to your microbial biochemistry, where you will be able to see uh, how NADH and FADH, how they are being produced. Is it towards um, catabolic or anabolic reaction? So and so that we're able to also understand it along the line. So but what we are trying to tell you, I told you ever before that you should just imagine a bacterial cell. When you see a bacterial cell, they are not just ordinary cells, although they are microscopic, but everything, the future, their own fate, the fate of every microorganism depends on the cellular activities or metabolic activity that takes place inside there. Whether are they going to be able to balance or maintain or exist in equilibrium. So that is very very important. Now let us now talk about enzyme. This is time to talk about enzyme. Now enzyme lower the activation. You will not by this time around you will know exactly that enzymes are biological catalysts. So enzyme determines the speed of life. No biological reaction will take place in the absence of what? An enzyme. And that's why the activation energy, so enzyme lower the activation energy. Remember, it lowers the activation energy, but do not change the free energy, which is delta G or reaction equilibrium. So what we are saying in essence is that this is the enzyme, this is the active site, and you can see Okay, sorry, this is the protein, this is the protein, and this is the active site. Once this one binds to it, so let's look at this graph. This graph, you can see that substrate concentration, the substrate concentration of the enzyme, you can see, but the velocity is this one. So there is going to be a level where v, which is velocity, is going to be the v max, v maximum, the point at which the velocity is high all over the substrate. Okay, okay, all over km plus s. Okay, so we are going to, okay, km reflects substrate affinity in nutrient environment, allow this and this one. So to say, so that is a substrate. You can see s here, that is substrate, which is this. We talk about this, I think, in the next slides. So you can see now there are three different types of enzyme. There are three different types of enzyme. Okay, don't forget, during glycolytic reaction, how are they being, I mean, how is glycolytic reaction, or all these metabolic pathways, how are they regulated? They must be regulated in the sense that, for example, if glucose is will continue to be converted from glucose to pyruvic acid indefinitely, you will see that at a point in time, you will see that there's not going to be any glucose that will be left. And the pyruvic acid is also going to be so toxic to the level that at the end of the day, even the cell, it is the product that will even kill the cell. So what we are saying in essence that is cell must continue to maintain a state of equilibrium. State of equilibrium. So there are three basic types of enzymes inhibition. So you can see this one is binding to the active sites. That is called the competitive. It is competing for the same active sites as the substrates. Okay, so inhibitors, it is this one. This is an inhibitor. Inhibitor is a kind of enzyme that will, you know, under normal circumstance, when we talk about enzyme we go, or we move towards making progress. Okay, that is forward reaction. But there are also certain inhibitors. So enzymes, that one will be positive, but that of inhibitor will be negative. That is, the chemicals that serve as enzyme that will hasten the rate of chemical reaction or rate of biological reaction. There are some that must also bind to them in order to stop that kind of reaction. Just a typical example of that is what I've just mentioned in the case of all these cellular pathways, whereby after some time the cell must continue to use what is called feedback inhibition. Okay, yeah. So whereby so example of an enzyme or inhibitors that will compete with the same active sites. So it's just like it is the first one, first come first serve. It is first one that gets there that is going to perform the task. So inhibitors is competing with the same active sites. That is competitive inhibition. Competing with the same active sites. That is what? Competitive inhibition. The second one, and this is the graph. You can see the graph. You will see that at this point, at a point, two of them. This is one is for inhibitors. You can see this one is for the inhibitor. Then you can see this one, which is for the enzyme. They are competing for the same active sites. So you can see this is the velocity, this is the substrate. So you can see that at a point in time, the two of them, two of them now meet. That is competitive inhibition. The second one, which is non-competitive, this one, inhibitors might be competing for the active site, whereas the enzyme is going to compete for another. So you can see. So at no point would they meet. You can see the difference. At no point would they meet. So inhibitor will bind at allosteric sites. This is the allosteric site, whereas this one is going to bind at another. So it's like a journey that you can never miss. Okay. Now the third one is uncompetitive. Uncompetitive. This one is non-competitive. Why it is non-competitive is just because it depends on which one comes first. These two, which one comes first? That's what they are saying. If it is the individual that binds to this place first, that means the chemical reaction will do what? Will move forward. But if it is the enzyme that now binds at the end of the day, that means it's going to be forward. Now, this one is non-competitive, but they are still what? Bind. They are still struggling with anyone that comes first. If it is this one that binds first, fine. And if it is this one, so but at no point will they have any they will not meet, no meeting points. Okay, so that is very, very important. Okay, now the third types of enzyme inhibition is what is called uncompetitive. From the onset, this one is going on its way, this one is also going on its way. So you can see. So this one has its own different binding sites. Inhibitors has its own binding site, and also the enzyme, and the other enzyme also has its own binding site as well. So that protein will have a binding site for inhibitors, then it's going to have another site for the enzyme to also bind. Okay, that is very, very important. And example of catabolism, which is a building up, I've told you, is glucose down to Krebs cycle. You will remember that glucose is going to start from glucose, then to pyruvic acid, then from pyruvic acid, malonic acid, and so on and so forth, until it now becomes what? So at this point, this is from here to here is anaerobic respiration. Anaerobic respiration. Then from here to this place is aerobic respiration. I'm very sure they must have learned that in microbial biochemistry. So we might not be able to do more or explain more. Although I am going to also make a different video for where we will explain catabolism or catabolic reaction in full. So I remember I also gave us some short ways to be able to crack, so that you don't have to cram everything. There are certain shortcuts that you'll be able to know what are those compounds that will be found at every point in time. But those forget that in respiration, glucose is very important, and it is the starting product, the raw material for both anaerobic and aerobic. So TCA cycle, pyruvic is at the center, is at the center, is at the the pivot for all cellular respiration, be it anaerobic or be it aerobic, even fermentation as well, because it's going to be from here that this one will now proceed to it's going to proceed to ethanoic acid, to ethanol, yes, sorry, to ethanol for as a fermentation product. Okay, so now metabolic switching, I've told us. So the three stages or the three strategies of energy, how do they get energy and how do they make it available. Now, in in aerobic respiration, that has to do from glucose down to where oxygen will be released. You see that ATP is going to, the number of ATP that will be yielded is going to be between 30 and 38. So this is the highest form of efficiency. So that is how the cell, that we are still talking about microbial metabolism. That is how the cell will be able to acquire, and that cell that is able to liberate or get up to between 30 and 38 is operating at its highest level of efficiency. Then the anaerobic, their own final acceptor, instead of the final acceptor of aerobic to be oxygen, their own is nitrates, sulfates, and CO2. Now, what happens is that moderate ATP will be generated, which is very, that means they are very flexible. They have flexible survival strategy, especially in the absence of oxygen, that is in anoxic environment. Then the next one, I told you the gateway is a pyruvate acid that has to, from pyruvate it will go to ethanol, and that is for fermentation. So the final acceptor here is organic ethanol, is organic molecules, and this one does not need etc at all. They do not need it. So, and at the end of the day, only 2 ATP via substrate level phosphorylation. So, and the primary purpose of this fermentation is to be able to regenerate NAD plus, to be able to keep their own glycolytic or glycolysis running. So what we are saying in essence is that microbes actively switch. They can switch depending on the environment, depending on what are the surrounding, the enabling environment around them, and that is what is called thermodynamic efficiency. So ability of any microorganism to be able to switch in between the strategies whenever oxygen is available. Sorry, if they have the necessary raw materials, they will go into what, aerobic respiration, whereby they will generate more of ATP. That is when they will need it more. So, and in aerobic respiration, we just generate moderate ATP, sometimes between 4 and 6 ATP. If that is what they need, they will go through anaerobic respiration. And fermentation as well. So that is when they need just when the currency, the energy currency that they need is not too much. So what they will do is they will prefer to undergo the process of fermentation in terms of their respiration. So the only thing is that their final acceptor, they know that it's going to be organic molecule, which is going to be ethanol. So that is what's going to. But what we are saying is just that the survival level of any microorganism will depend on their ability to maximize their thermodynamic efficiency. Okay. Now this is anabolic and amphibolic up. Okay. Don't forget, anabolism is reductive. Anabolism is reductive, precision regulated, and highly energy consuming. Then the duality of mechanism, central pathway like a TCA, you remember TCA, okay? That is a Krebs cycle. TCA, like TCA are amphibolic. Why are they amphibolic? Number one, you will see them that they will be providing at a point in time, they will be providing, they will be building some. You can see they are building one part, they are building amino acids. At a point in time, they are building lipids. At a point in time, they are building what? Nucleotides. Okay, and at a point in time they are building this. Then at the same time they are also catabolic. So that is why they call them the duality of metabolism. The duality of metabolism is when you have uh, a specific cell or a specific reaction that is both catabolic as well as both anabolic. Some of the microorganisms that has the ability to undergo this duality of metabolism, I think it is something that is going to be the subject of discussion for some other time. So then, what are the cost of building? Biosynthesis require massive investment of ATP and ADP. So under normal circumstance, the type of house you are going to build will depend on the amount of money, currency that is with you. So what we are saying in essence is that the cell, what determines what a cell will do in the presence of unfavorable condition will depend on the available energy, available ATP, available currency that is there. So if the environment is such enabling, that means the cell will perform very well. The thermodynamic efficiency is going to be too high. It's going to be so high, and you will just see that they will use that for their structural integrity. So, but once the available energy, just like when there's no money, you just, even if you want to, if you must build any house, just build any house. So that is just the analogy about it. Now, I want us to explain and see growth as a physiological expression. Don't forget where I have been coming from. I told you that this course has two branches. The first branch is microbial metabolism. That has to do with cellular activities. The second one is physiology. So all what we have been doing before this from this place, this is talking about metabolism, microbial metabolism, microbial metabolism. This is also microbial metabolism. This is also microbial metabolism. This is microbial metabolism. This is microbial metabolism. This is microbial metabolism. So the second aspect, which is a microbial physiology. So, and we want to consider growth as an example of that. Now, looking at this, all of us, we are familiar with this because that is where our journey as a microbiologist should begin. We have the growth. The growth curve is divided into four stages. Number one, we have the lag phase, followed by the log phase, followed by stationary, and followed by the death phase. So in some textbook, at this point, they call it exponential. Call it exponential because that is when you have almost irreversible division or multiplication of the number or the population of that organism. So without wasting our time, you see that it will start from here, okay, and that is the level of adaptation. They will begin to get used to the environment. There's going to be high metabolic activities that is going to involve what is called the enzymes that they will need. So it's just like it's going to be the preparatory stage, just like you want to embark on a journey, and when you want to embark on that journey, you have time to start arranging your load. No movement yet. You check your vehicle, you check the water level, check the oil level, check everything level. But at that point in time, there's not going to be any movement. So that's why at this point, which is the lag phase, no division, no division at all. Now the next thing is the log phase. This is the level at which there will be maximum metabolic efficiency. There will be increase there's going to be, uh, let me say, exponential increase. There's going to be increase in the number, rapid cell division, maximum metabolic efficiency, and most sensitive to antibiotics. So at this point, because they are at this growth level, so at that level, antibiotics will definitely have effects on them. Okay, then they will now enter the third phase, which is called, that is the stage at which all the nutrients that is available for them in that medium is already getting exhausted. So it's just like somebody, you are going on a journey, and that journey you get money, you get food, you pack everything, almost everything, and at the point in time you start moving, moving until you get to a level whereby your food is getting exhausted bit by bit. Your energy is getting exhausted bit by bit. So what you just discover is that the nutrient is depleted, then waste accumulated. Waste is going to accumulate, then stress response will now be activated. So at that point in time, the number of the deaths will be equal to the number of what? Of the growth rate. So that is why they call it stationary. So if 20 are dying, 20 will be growing as well. That is the growth. So until they now enter the fourth, which is the final stage, that is the point at which all their nutrients is already depleted, is already exhausted, and the next thing is that they will continue to die. The number will now continue to reduce like that. So, and they call it a metabolic collapse, the death phase. A phase whereby there's no more cellular activity. A phase whereby there's nothing like a functionality any longer. So the cell will not gain energy. The cell will be losing energy from time to time. It's just like the time of old age. And at that old age, you just see that everything is becoming wasted. Everything, so just like that. So the number no more grows. What you see and what is going is going to dominate is going to be death and death and death until the final stage. So the environmental negotiation and community physiology. So now, what are those important environmental conditions that will have effects on the behavior or on the metabolic activity of any microorganism? So number one, increase in temperature. As you can see, increase in temperature, the pH, then oxygen. So that is a stress adaptation. So once all these three environmental conditions they are prevailing, that means microorganism must look for a way to alter, they must be able to adjust their metabolic pathway in order to be able to survive. So that is what we are saying. E.g., hemiophisious adaptation alters membrane lipid composition to survive temperature. Enzymes like catalase detoxify oxygen. So it's part of their coping strategies. Part of their coping strategies in order to be able to survive the very unfavorable condition. Then another one is quorum sensing and biofilms. So sometimes, apart from seeing microorganism, the ability to be able to cope with favorable condition that could be as a result of an increase in the temperature, pH, as well as oxygen. So there is something we call quorum sensing and biofilm. So when population reach a density threshold, chemical signal will alter the gene expression collectively. So what we are saying in essence is that you also give rise to what is called biofilm. Biofilm is the defense, they use it as kind of defense that all the microorganisms in that medium will now come together to form a kind of a community that will be able to protect it. Just like when you are in your area and maybe you have attack of you have armed robbers that wanting to liberating you and they want to come and visit you. You now, all of you will now arrange, you now form a kind of a vigilante collecting this in order to be able to protect your own community against infection. So the same thing that they do. So all these microorganisms, they will come together irrespective of their species, irrespective of their type, irrespective. So they will come together and form what is called a biofilm. Biofilm will now protect them and defend them from the external aggression. So just like I told you earlier on, that under normal circumstances, there must be well, even when you are building up, when there is energy generation, you cannot just continue to put or build on energy without having a regulator. So all the cells must have a regulator, that is gene control. So and that's the reason why you must see this as an example. So this region is called a promoter region, this region is called the operator region, and this is the key, which is the master key, and this is a repressor, which is a protein. So at this point in time, these are the structural protein, like Z, like Y, like this. So this one will be elaborated when we are in a different video, when we'll be talking about microbial genesis. Okay, microbial genesis. I think that's one of the first or foremost topics in microbial genesis. But the logic behind it is just when you get home, when you have electricity, you are going to have a switch. A switch is where you will turn it on when you need the light, and you turn it off when you don't need it again. So that is what is called the gene or genetic control or regulator, and that is an example of where we have the indissoluble enzyme that lack operon, and also have a tryptophan operon. So more of this we are going to talk about it later on. Now, applied microbial physiology. Where can we apply? Where can they be applied? So where can they be useful? So it is useful in clinical targets like antibiotic targets for this. I won't be able to, because of my time, I won't be able to go into detail, but I just want you to know that it's very, very important in 4 key areas: clinical targets, industrial fermentation, metabolic engineering like CRISPR. They just discovered that microorganisms, they now have some certain gene whereby they can do what? They can repair, they can fix. If one is bad, the other one will come and repair it. Okay, so then we also have, it's also very useful in the pathology, that is in disease control. Okay, now just like I said, this is a Edu-Interact framework. First thing is we should be able to understand, we should be able to define core mechanism. You want to have a good glimpse of this. You should have understand. You must understand. You visualize. You apply. Then you optimize. So the final takeaway is that microbial physiology is not an abstract chemistry. It is the strategic computational logic of life. And just like I said, under normal circumstance, the world is now being digital. Now, if you want to learn about all these metabolic products now, you now have some databases where you can go and you'll be able to study everything that happens in the whole cell. You will conceptualize it and be able to see and be able to go forward on that. So having said that, I think this is a very good area to stop today. Don't forget what you have learned. I was able to break down the two very important concepts in this course, microbial metabolism and physiology. And we were able to look at all this as a metabolism. Then we also talk about growth as a physiological expression. We also talk about how microorganisms are able to negotiate their environment as a result of unfavorable condition. Then we also look at how all these being controlled, gene off and gene on, which I said we will learn more about this in microbial genetics. And we also look at what are the importance or what are the applications of microbial physiology, so that some of you will not continue to say, why are we doing all this? So they are very, very important today and very useful to us in four key areas which we have mentioned. And just like I said, this platform or this ecosystem is a new is a kind of a paradigm shift from our traditional mode of learning to the digital way of learning, which has to do with understanding the basic concepts